All right, moving into chapter 6 now, looking at bones and bone tissues. First of all, what are the parts of our skeletal system as we move into mainly, of course, thinking about our bones and what this is focusing on, but also part of the skeletal system includes cartilage, and there are 3 different types of cartilage: hyaline, fibrocartilage, and elastic. And then we also need those tendons and ligaments that help us attach some of the muscles to the bones. So what about the functions of our skeletal system? And most of these are probably things we could think of on our own. Definitely support. Of course, bones are really hard and rigid, but also the cartilage provides that flexibility. And then ligaments help connect the bones to each other, and our tendons connect muscle to bone. So protection, we think about the skull protecting the brain, ribs protecting our thoracic cavity and our lungs. We need, of course, the skeletal system to be able to move based on those muscles that attached, again, using those tendons. But bones also store, especially calcium and phosphate. So we think about like osteoporosis and bone health. So we can use them to store up these molecules. And also adipose tissue is stored in the marrow cavities as well. And then also blood cell production. So probably these last two you may not have quite instinctively went with, but storage and blood cell production definitely important parts. So our bone marrow is important to giving rise to those blood cells and platelets. All right, so focusing on cartilage to begin with, and then we'll get more into the bones. So hyaline cartilage is the main type. Again, we also have fibrocartilage and elastic cartilage, as we just mentioned. Couple terms here: chondroblast and chondrocytes. Coming up, we'll have osteoblast and osteocytes, similar. So chondroblasts form the matrix of that cartilage that we'll see here in a minute. And then the chondrocytes are around that matrix and within the lacunae. Then also the matrix there is made up of those collagen fibers that gives that strength, and then the proteoglycans for resilience. Now there's also a perichondrium, and that's double layer CT for connective tissue. So that's going to cover the cartilage except in articulations. So we have an inner and an outer part. The inner is more delicate, so we think about kind of protecting it on the inside. A few less fibers, and it contains those chondroblasts that make the matrix. And then the outer part is where it's going to be innervated with the blood to give it nutrients, as well as the nerves to penetrate it and activate it. No blood vessels, though, actually within the cartilage itself. So it's just on that outer exterior. The cartilage itself does not have any blood vessels coming into it. Now, articular cartilage, this covers bones at the joints. So it does not have any perichondrium growth. So there is the appositional. These are the new chondrocytes and the new matrix that are out at the periphery, so kind of the outside, and then the interstitial, so think about it being on the inside or interior. So this is where those chondrocytes with the tissue divide, and more of that matrix is going to occur between the cells. This is what the hyaline cartilage looks like underneath a microscope. So if we were visualizing it under the microscope, again, the chondroblasts are more out on that periphery, and then the chondrocytes are more on the internal part here. So that interstitial growth that's occurring, that appositional growth, more on the outside part of that. And then the perichondrium all the way out there on the periphery where those blood vessels and nerves would actually attach to. These other parts we'll be seeing later on. So as we look at this developing long bone, and then the difference between young and adult bone. So we'll be coming back to those later, but just to kind of see where it all fits in together. Now, what about the bone histology? So what does bone look like under a microscope or looking at the tissue itself? So it's about 35% organic, so carbon and hydrogen containing, that's that collagen and the proteoglycans, but it's actually majority of it is inorganic with a compound called hydroxyapatite, which is calcium phosphate and some hydroxyl groups, so these crystals that form. So if that mineral is removed, then the bone becomes too bendable, it's too flexible. If the collagen is removed, then the bone becomes too brittle. So again, both the mineral and the collagen are needed in order for the bone to be not very bendable and also not too brittle. So it's very strong. Now, what about the bone cells? So I mentioned we'd come back to these, the blast and the clast. So osteoblast, these are going to form the bone through a process called ossification or osteogenesis. And here the collagen is produced in the endoplasmic reticulum and then the Golgi. So if you remember back to your bio, the ER produces molecules, the Golgi kind of modifies, sorts it, and sends it where it needs to go. So the collagen gets made in the endoplasmic reticulum, shipped to the Golgi, and then it's released from the cell through a process called exocytosis. And then the precursors for that inorganic material, the hydroxyapatite, that's stored in vesicles within the cell and also gets released through that exocytosis process. Now, the process of ossification is a formation of bone using those osteoblasts. So they communicate through those gap junctions. So you can think about the little tunnels between cells where they can share their cytoplasm. And then themselves are surrounded by a matrix. Now, osteocytes, so we have osteoblasts that are making the bone. We also have osteocytes. So this is in the mature bone cells. It's also surrounded by a matrix, but it can make small amounts of matrix to maintain it. So again, we have those lacunae, which are those spaces that are occupied by the cell body of the osteocyte. And then the caniculi, those are going to be the canals that are occupied by the cell processes. So again, the caniculus that are here, kind of the branching parts, so if you think about, kind of looks like almost like a neuron. So this would be like the dendrites and the axons there. And then the lacunae, they are the actual where the cell body is going to be. Now, what's going to happen here, again, we don't have a lot of nerves and blood vessels coming in, so the nutrients diffuse through tiny, tiny amounts of liquid that surround the cells. That's going to fill that lacunae and the caniculi, and then they can transfer the nutrients from one cell to another using those all-important gap junctions. All right, now our osteoclast, instead of building the bone like we saw with our osteoblast, so osteoblast build, so B for blast, B for build, osteoclast, they reabsorb or break down, so like clatter is how I remember it, so it's breaking down the bone or reabsorbing it. So this produces kind of a ruffled border. This is where that cell membrane borders the bone and starts getting reabsorbed, or we get resorption taking place. What's also happening here, those hydrogen ions get pumped across. Of course, hydrogen ions are associated with acid production, and acids typically break down or deteriorate things, and that's exactly what happens. It eats away at the bone based on those hydrogen ions. Then there are also enzymes that get released along with the hydrogen ions that digest and break down those pieces that are being released. They're derived from monocytes, which is part of our immune system, and those are formed by the cells in our red bone marrow. Then these multinucleated, just meaning lots of nuclei, usually cells only have one nucleus, but these have multiple nuclei, and they probably arise from the fusion. So they start coming together, cells start fusing together, so now each one brings its nucleus, so we have multiple nuclei inside of one cell. And then one other cell type here, stem cells, which you may have heard of. These are cells that can give rise to any type of cell. So this term mesenchyme, these are osteochondrial progenitor cells, just meaning progenitor, kind of the precursor. So they're making lots of different cells. So stem cells are cells that can become any type of cell. So this mesenchyme are the precursors to those chondroblasts or to the osteoblasts. So either one can be made starting from this mesenchyme. Now, what do these osteoclasts look like? Again, these are the ones breaking down the bone. So again, we kind of get this ruffled border, just kind of this invagination. It's kind of like little villi or little projections sticking off there, but this ruffled border here, it's going to be releasing the hydrogen ions that are shown here, or different enzymes that come out. And essentially it just eats away here at this bone that's here. Again, we have multiple nuclei because multiple of the cell types start coming together and fusing, and each one brings its nucleus, so it creates kind of a larger cell because of all of them coming together and fusing. Now we have what's called woven or lamellar bone. So the woven bone, this is where those collagen fibers are randomly oriented. So it just kind of looks woven together. This is the first type that's formed by the osteoblast during the ossification, so during that process. So this will be during fetal development, growth, but also fracture repair. So if we're fixing bone, this woven bone is initially made. And then we have this remodeling process where those osteoclasts remove the old bone that's there, and then the osteoblasts are going to build the new. So the woven bone is remodeled into lamellar bone. And that lamellar bone is the mature bone. So we can kind of think woven as being initial or kind of immature bone. That mature bone, the lamellar bone, is made from sheets of that lamellae, hence the name. And these are fibers that are oriented just in one direction. So instead of kind of being randomly all over like the woven, these are all going in one direction, but in each layer, but different directions in different layers. So on one layer, we may be going in this direction, but then in the layer above it, they would be going maybe in the opposite direction. All right, now we're talking here about spongy bone. Trabeculae is this interconnecting rods or plates of the bone. So kind of like a scaffolding is really what we think about. So this here is what we see. This is the trabeculae. So it's referred to as spongy bone because it has all these holes or opening here. So we have these spaces, but it's covered by what's called endosteum, and it's oriented along different stress lines. So this is showing under the microscope what these look like, and we'll be looking more at this here in a minute of what these cells actually look like. But these trabeculae are made up of these osteoclasts and osteocytes that are here, but ultimately they form kind of the spongy or wholly space-filled type of bone called the spongy bone. Now, compact bone, as the name implies, this is going to be solid, more the outer layer that surrounds each bone. So it has more matrix and is denser than the spongy bone, so it's more packed. We don't have all those open spaces in there. Now, the functional unit is called an osteon or the Haversian system. So these osteocytes are sandwiched in the ring like we had just seen of the matrix, kind of looks like a tree trunk almost, and then connected by those caniculi. There's like projections coming off of the cell body. So the central canal or the Haversian canal, this parallels to the long axis. The lamellae, that's concentric, so in circles, circumferential, and interstitial, so more on the inside. And then we also have the perforating or Volkmann's canal that's perpendicular to the long axis or to the central canal, so going in kind of opposite directions there. But both the perforating and the central canals have blood vessels. So the canal, we think about like water running through it. Well, in the case of bone, blood is running through those. So that provides the direct flow of nutrients from the vessels to those cells of those osteocytes within the caniculi, and then also from one cell to the next. And this is just showing, again, kind of our little tree trunk here. This osteon is that functional unit here. So they build on top. So we have this central canal in the middle, as the name kind of implies there. But then we also have these concentric lamellae that are here that are going in parallel directions. And then the Leucinia that is there as well, that's going to contain the osteocytes. Again, the osteocytes are going to be the ones in charge of those bone cells. And then just kind of looking on a bigger layer, again, here's that trabeculae, and then we have that compact bone, so our spongy bone, and then the compact bone. that's there. And again, those osteons are part of that compact bone that all come together. Now, a little bit more about that individual unit, the osteons. Again, they have that central canal, as we had just seen, right in the middle where those blood vessels go. The concentric lamellae of the bone that's surround that central canal. The lacunae and the caniculi contain the osteocytes and different fluids. Then the circumferential lamellae, that's on the periphery of the bone, and then interstitial, or on the inside, lamellae, that would be between the osteons, and it's remnants of those that are used and replaced through remodeling. All right, now that perforating canal, again, delivers the blood to the central canal, runs perpendicular to the length of the bone, so the nutrients and waste travel to and from the osteocytes using interstitial fluid. And then it also comes from the osteocytes and from one osteocyte to another using those gap junctions. So it can move through the lacunae and the caniculi, but also from cell to cell. All right, now moving into the bone anatomy. So the parts of the bone, starting first talking about the long bone, which is the bone we normally think about. So has the two ends, the two heads, not talking about like the skull or something. So the structure of a long bone, we have the diaphyse and the epiphyse. So the diaphyse is the shaft that's mainly made up of that compact bone and then just has a cavity on the inside or open on the inside. That opening or that cavity is called the medullary cavity. In children, that cavity has the red marrow for blood cell formation. In adults, the red marrow is replaced by yellow in the limb bone. So that does change as we get older. Now on the end, the epiphyse, or again, epi means outer. So this is the end of the bone. So that's primarily the spongy bone that's covered by compact. So the whole bone is covered by compact bone. It's just in the diaphyse, there's that cavity for the bone marrow, whereas at the ends, we have the spongy bone. And then within joints, it's covered with hyaline cartilage that's called articular cartilage. Now the epiphyseal plate, that's our growth plate. Again, hyaline cartilage is the main cartilage we're kind of focusing on, and it's present until growth stops. So once growth is complete, that cartilage is gone, and it produces what's called the epiphyseal line. So that's where the plate now becomes ossified when the bone stops growing. We also have the periosteum. This is the connective tissue membrane that's covering the outer surface. So all the way on the outside that compact bone. It's the outer portion that has dense, irregular collagenous connective tissue. So it does have innervation from the blood vessels and from the nerves. The inner is a single layer of bone cells that has the osteoblast and clast, and then also the progenitor stem cells. Now, fibers of the tendon and ligament become continuous with the fibers of the periosteum. And then we also have these perforating fibers again here. Here they're called Sharpey's fibers, based on who kind of talked about them. So some periosteal fibers penetrate through the periosteum and into the bone, and that's what these fibers, so they perforate, they move through the outermost kind of connective tissue into the bone. So this strengthens the attachment of the tendon to the bone. Periosteum on the outside, and then endoosteum on the inside. This is a single layer of that connective tissue that lines the internal space, including those cavities in the spongy bone and the medullary cavities, so any openings. And this has osteoclast, osteoblast, as well as those progenitor cells. And this is just showing our long bone here. So again, here at the ends, the epiphyse, then the diaphyse is the shaft there. Again, this would be an adult, so this is that yellow marrow that's in here instead of the red bone marrow or the red marrow. So again, the diaphyse is the shaft. At the end, we also have the epiphyseal plate that's there as well. Then as far as within each of these, we have, again, spongy bone that's there at the end and the epiphyse. The periosteum that's on the outer part. The endosteum is on the inside there, the inner layer of that cavity that opens up here, that medullary cavity. Then if we just kind of peel away the layers, so that peristele, again, blood vessels coming in there. We have these perforating fibers that actually move through the cartilage and into the periosteum there. Again, our osteons with that central canal right in the middle. And then all the way inside here would be our spongy bone, compact bone on the outer part. Now, a few different types of bones that we have here. So we already talked about the long bone, but we have flat bones. There's no diaphysis, so there's no shaft to it, or epiphysis, so there's no like end and middle. It's just sandwiched, just spongy between compact bone. So what we see here is just a bunch of spongy bone, and then we have compact on the top and the bottom. We also have short and irregular bones, which is compact bone around that spongy bone, similar in structure to the epiphysis of the long bone. But there's no diaphysis, so no shaft, and is not elongated. So some flat and irregular bones have what are called sinuses, which are air-filled spaces that are lined by mucous membranes. All right, so how does our bone develop? So we'll talk about intermembranous ossification and endochondral ossification. So the intermembraneous takes place during in the embryonic connective tissue. Endochondral is in the cartilage. So both of them go through ossification, as the name implies there, to make that woven bone initially that's then remodeled into that lamellar bone after the remodeling that takes place there. So looking first at the intermembranous ossification. So again, in the connective tissue membrane during that embryonic mesenchyme. So this is going to form the skull bones, part of our mandible, and then the diaphyses of the clavicle. And this actually starts in the 5th week of embryonic development. So again, that mesenchyme is the progenitor, gives rise to those stem cells. So it becomes condensed in the brain to form the membrane of the connective tissue, but it has these delicate collagen fibers. Now the centers of ossification are in the membrane where ossification begins. And then in the embryonic development, and even initially once the baby is born, are the fontanelles. These are the large membrane covered space between the developing skull bones. So it's unossified, it's not hardened or calcified. So this is the soft spot on an infant. So this is to allow the brain to develop and grow instead of being locked in one shape. Now this intermembranous ossification, so again, the osteoblast formation, so some of those embryonic mesenchymal differentiate into osteochondrial progenitors, and then ultimately give rise to those osteoblasts. Then the osteoblasts form the bone matrix that surrounds those collagen fibers of the connective tissue. And then once they're embedded in the bone matrix, now the osteoblasts become osteocytes, and they're going to start being used as reabsorption. So as a result, we have many trabeculae of woven bone that get developed. Now the spongy bone formation, now additional osteoblasts gather on the surface of the trabeculae. They produce more bone, and thereby causing the trabeculae to become longer and larger and longer. And then it forms the trabeculae joined together in this interconnected network that's separated by spaces. So again, we can see these spaces that are forming here between them. Now the compact bone formation, this is the cells within the spaces of the spongy bone form the red bone marrow. And the cells surrounding the developing bone specialize in forming the periosteum. And those osteoblasts, again, B for building, from the periosteum lay down bone matrix to form the outer surface of the bone. Then the end products of intermembranous ossification are bones with outer compact bone surfaces and spongy centers. And remodeling converts the woven bone to the lamellar bone and contributes to the final shape of the bone. Now the endochondral ossification, now this forms bones of the base of the skull, part of the mandible, the epiphysis of the clavicle, and most of the remaining bones of the skeletal system. So cartilage formation begins about the 4th week of development, and some ossification beginning about week 8. Some doesn't actually begin until 18 to 20 years of age, depending on the bone we're looking at. So this process here, again, in the first part, the chondroblasts make the cartilage model that surrounds the periosteum, except where the joints are going to form. Then the perichondrium of the diaphysis becomes the periosteum, and the bone collar is made. So internally, the chondrocyte hypertrophy, so they start deteriorating and becoming smaller, and calcified cartilage starts to form. Then a primary ossification center forms as those blood vessels and osteoblasts invade the calcified cartilage. The osteoblasts lay down bone matrix, forming spongy bone. And then finally, the process of bone collar formation, that cartilage calcification and spongy bone production continue. So calcified cartilage bones begins to form the epiphysis. And a medullary cavity begins to form in the center of that diaphysis. And this just continuing on here, then in the mature bone, the epiphyseal plate has become the line as it becomes calcified or ossified, that ossification, except at the articular cartilage that's become bone. So then the original cartilage model's almost completely ossified, so the cartilage, that hyaline cartilage that stops after production of the bone, it then becomes calcified. And then the unossified cartilage becomes the epiphyseal plate and that articular cartilage. And then finally, secondary ossification centers form in the epiphysis of the long bone.